Dr. Sneha, who is faculty at the Department of Microbiology, Bangalore Medical College and Research Institute. She works on projects in areas of sexually transmitted infection, rapid identification of bacteria, sepsis, escape pathogen, dried blood spot for HCV diagnostics, community acquired UTI. Thank you, madam, for giving your time, and please take the stage.
A very good afternoon to one and all. And I would like to thank the organizers for this opportunity to present our work and share our thoughts with you all. And also apologies because my slides will not be in the, as colorful and as presentable because I have prepared it at a very short notice. I'll be talking about application of nanotechnology or rather nanobiotechnology in healthcare. And I would like to share our own experience with you, what we have done. So basically, I'm a diagnostician, I'm a microbiologist. We help in diagnosis of infectious diseases. And I work with Bangalore Medical College and Research Institute, which is a premier government institute. So I will just skip the slides in the interest of time because we have already heard about what nanotechnology is and how it is helpful not only in diagnosis but also in preventive aspects as vaccine delivery systems and also as therapeutics in case of viral infections. Just we heard Dr. Jayaram's lecture as to how they help in antiviral drugs also. Of course, a lot of work is happening in this field. And when we talk about human health, as we all have seen, in addition to cancer, infectious diseases are the one which not only affect individual, by causing a lot of morbidity and mortality, but also they cause extreme clinical calamity and affect the socioeconomic activities globally, as we have witnessed during the recent global pandemic. And in case of infectious diseases, rapid diagnosis is a very important thing. And though there are a lot of platforms available, application of nanotechnology-based detection platforms for rapid and sensitive diagnostics is the need of the hour. So integration of these nanoparticles or nanostructures with a molecular recognition in whatever way we can is having a lot of scope in diagnosis. In this regard, there are a lot of platforms available, such as antibody recognition or aptamer detection, phage-based sensors, and even colorimetric methods. And also, there are some publications based on the human olfactory system as a method to, detect these. So, what are these aptamers? I'm not sure if everybody is aware of, so I'll just give a brief, idea about what these aptamers are. So these aptamers are single-stranded nucleic acids, either DNA or RNA, which are selected against any specific target. It could be a target which is of interest to you. And this specific target is selected by a repetitive or iterative rounds of selection of a process called as SELEX, which is systematic evolution of ligands by sequential enrichment. That is called as SELEX, right? This is a procedure which helps in choosing the right aptamer. And these aptamers have advantages because they are very small in size. As we saw, it's just a strand of DNA or RNA there. And because it is small in size, it has low immunogenicity, it has very good tissue penetration, it has longer shelf life, and as it is stable, it has obvious advantages. And in addition to detecting small molecules, it can even detect the whole organism. And because it can change itself into a tertiary structure, it can bind with the targets with very good affinity, and also when you develop the platform will have very good sensitivity, specificity, and can be used as a point of care test, or what we call as POCT. And for any POCT to be useful in the clinical field, it has to satisfy WHO criteria called as assured. That is, it has to be affordable, sensitive, specific, and also user-friendly. Apart from that, it needs to be robust, to be able to be used in harsh field conditions, and equipment-free. It shouldn't depend on any specialized equipments, otherwise it fails the purpose, and it should be deliverable. So this is the criteria which any POCT should satisfy. So there are a lot of platforms available, and I would like to share in about next 5 minutes as to what work we have done with aptamers in our lab. And if you just Google and see, a lot of publications are already existing. And with respect to infectious diseases, as you can see, there are a lot of point-of-care detection tests for detection of E. coli from water using gold nanoparticles. And then there are a lot of nano antibiotics for AMR, for detection of antimicrobial resistance and also to overcome the same. There are aptamers to detect pathogenic bacteria. So in this regard, we got a funding from ICMR for a project on Vibrio cholerae. Cholera, as you all know, is a disease, a bacterial disease caused by consuming contaminated food or water, and it is caused by Vibrio cholerae, which is the organism causative agent. And this disease, as we all know, can start as an outbreak, can spread rapidly, thereby leading to epidemic and even pandemic. We recently had one outbreak in Bangalore in our own hostel as well. So this is a very important bacterial pathogen, and though there are a lot of tests available to diagnose it right now, right from a simple microscopy to culture to molecular tests, each has its own disadvantages also, right? So, and WHO in its Sustainable Development Goals aims to eliminate cholera as a disease by 2030, and rapid diagnostics is a key structure in that. So in collaboration with Indian Institute of Science and AIIMS BB Nagar, we undertook this project with the objective to develop a rapid assay to detect cholera toxin of the Vibrio, because not all Vibrio are pathogenic, and detection of the pathogenic Vibrio cholera is very important. So we aim to detect the cholera toxin. But just keeping the device ready is not very helpful. It has to be of use in the clinical field as well. So we also had the secondary objectives to test it on culture isolates, but as has been discussed since morning, unless it is useful on a clinical sample, the whole purpose fails. So you need to develop an assay which can detect the organism in the clinical sample. So we also had aim in mind that we have to test the assay on the positive and negative stool samples. As you know, cholera is a disease which causes diarrhea, and stool sample is the one which is helpful in diagnosis of the disease. And also the test needs to be validated so that it is market ready. So just briefly about the methodology. So we isolated the Vibrio cholera from the clinical samples received in our lab, characterized the isolates, confirmed that it is indeed Vibrio cholera, and also we stored the stool samples for usage later. And regarding aptamer selection, we did not perform the selects ourselves. We used the aptamer which was already available in the literature using the selects technology. So this is the reference article from which we chose the aptamer. And for the cholera toxin B, initially we started with the commercially available cholera toxin B procured from Sigma Aldrich company. And before we wanted to go ahead with the platform development, we checked the binding affinity, and we found that the commercial CTB did not have a good binding when we tested with EMSA, electrophoretic mobility shift assay. So we cloned the CTB ourselves with the help of the colleagues, and we could successfully clone it, and also it showed good affinity by both STS page and EMSA. And then we took up the lateral flow assay development. So the principle based on the article of reference we used was to have a duplex dimer dissociation mechanism. So basically here, the aptamer with a specific affinity towards the target, in which in our case was the cholera toxin B, in a duplex format will bind with a partially complementary DNA which is tagged to a gold nanoparticle. So some part of DNA sequence is free while the remaining part is bound to aptamer. So in presence of the analyte or the target, the duplex will get dissociated, the bound part of the DNA will get free and gets hybridized to the sensor on the test line, thereby giving the test line. And I think I don't have to go into the details of lateral flow assay. The previous speaker has already dealt with that. And unless the control line comes, we can't say that the test is valid. So the control line has to show the color. And if the test line shows the color, it is positive. If the test line does not show the color, the test is negative. So here are few of the photographs. So this is the principle which I just explained. So the gold nanoparticle with a duplex aptamer which has a free DNA binding end. And once it binds to the analyte, it will dissociate, go and bind to the test line and bring in the color change. So, first picture here from our lab shows the development of the rapid assay using the cholera toxin which was cloned and used. So, it clearly showed some amount of color change there, so our test was working. But we wanted to test whether it detects it in the culture isolate also. So, the next picture here shows the testing of the developed assay for the detection of cholera toxin from the culture. Once you cultivate or grow the Vibrio cholera bacterium, it was able to detect the cholera toxin in the culture isolates also. So the third objective of testing it on the positive and negative stool samples also gave a good result, wherein we could see the control and the test spots on the positive and negative stool samples respectively. And we also tried to validate it using a series of stool samples, and here are our the photographs which show that test was valid and it showed positive result from the positive stool sample and negative from the negative stool sample. However, there were certain challenges because we were not able to give 100% results. There were some false positives and false negatives also. So we had problem with specificity and to some extent sensitivity. Sensitivity was more or less taken care of. Yeah, so I will just conclude here by saying that nanotechnology is definitely the field in which lot of diagnostics assays are being developed, and of course it will happen in future as well. Aptamers are one area of interest which can be taken up to bring in POCT tests. However, there are challenges as we also faced in our study, but they can be overcome only by enhanced communication and continued collaboration, because we doctors can provide the inputs and to some extent we can help in development of the kits, but we need the help or collaboration from the scientists to overcome the technical challenges, and interdisciplinary collaboration is the key. So I would like to thank for the opportunity and the time, and any questions I think we can take up later for want of time. Thank you.